Hi there. I'd like to talk a bit today about Benchling's vision for lab automation and a bunch of different use cases that we've seen with clients and labs that use us, ranging all the way from strain engineering to, in the past few months, a lot of work with COVID testing. So, quick bit of background. My name is Reed Molback. I'm a product manager at Benchling, and I manage our platform team as well as our lab automation team. And what we're going over today is kind of how Benchling is bridging the gap between lab informatics and hardware. And we're going to kind of talk through this in three stages. First off, we're just going to cover what Benchling is and what we've historically been good at, which is cloud-based informatics solution, tracking, platform R&D, stuff like that. The second section that we're going to cover is some of the trends that we've seen in automation across a lot of labs that we work with. And then finally, we're going to tie it all together with Benchling's offerings for lab automation and how we can really help. So to kind of start with, we have become, over the past several years, a trusted partner for life science leaders, in all sorts of areas. We work with hundreds of different companies. We work with over thousands of academic labs. We have, and if you look at the, types of science that people are doing, there's lots of therapeutics, lots of kind of cutting edge gene and cell therapies, things like that. But there's also a lot of other applications we see. Lots of synthetic biology, agriculture is now a focus. And most recently, something we didn't really expect to get involved in, but diagnostics, for a lot of, COVID testing that we've kind of needed to address, in the past several months. So really, what it boils down to is no matter what the area, our mission is to accelerate the pace of life science R&D. And the way we do this is, with what we call a unified and biointelligent cloud software. And what does that mean, right? Well, what that means is it's a suite of, very modern, snappy tools that you can use for your individual day-to-day work. Inventory software to, track your samples in your freezers. Molecular biology tools to design vectors, a lab notebook, kind of backbone of any of your experiments. But what really sets us apart is that, these are all individual tools that work really well, but they all sit on top of the same database. So, the vector that you design in our molecular biology tools is the exact same vector that you'll be tracking in our sample management tools and, that you'll cloud out samples of. It's the same record that you might request if you request some DNA to be amplified. It's the same thing that's going to be linked in the lab notebook when you're working with it. So no matter where you are in the platform, you're one click away from seeing all the different ways the data that you're working with is being used. That's something that you really can only get when you have kind of this unified platform. But what I want to kind of talk about a bit is that we don't just want to have a seamless integration across data in Benchling. What we also want is to be able to integrate Benchling with the rest of your R&D ecosystem. So, my job, I don't focus on any one of these applications. I'm not focused on the notebook or, the registry, something like that. I focus on how we can take all the data in that platform and integrate it in all these other ways. So if you need data for IP filings or regulatory submissions, how can you easily get that? If you need to extract the data to use it with your own third-party software, how can you get that? And what we're going to be talking about today is, when you want to use this data along with lab automation and instruments in the lab. What can we do for you there? So let's talk a bit about lab automation. Currently, we are seeing, over the past 5 years or so, there's been a huge trend towards higher throughput requirements. People want to test more samples, and they want to run more kinds of tests on samples. And this has really led to an increase in investment in robotics. If you're running 5 plates, if you're running 10 plates, you can have a guy sitting there pipetting it. It's boring, but it's doable. A lot of the labs we're working with are talking about tens of plates a day, hundreds of plates a day. And when you get to that level of throughput, it's a really bad use of time, to take your scientists who've spent, 10 years on their doctorate and, probably another 10 years in the industry, and having them just pipetting out plates all day. It just doesn't make sense. It's not a good use of time, and it's boring. So what we're seeing is people are, buying more and more robotics. But what happens along with that is you're generating more and more plates, you're running more and more tests, and you're getting more and more data. And the data doesn't actually, there's no easy way to get it into the informatics systems that you're using to actually track the samples themselves. So what we see is that there are all these big investments in the robotics and automation, but a lot of our customers aren't able to maximize those benefits because the lab informatics are disconnected from all that. If you look at more traditional limb systems, a lot of these are very rigid and hard-coded for a specific process, for a specific robot, for a specific assay. And this works great in a lot of contexts. If you are doing small molecule work, if you're running the same thing over and over and over again, these kind of rigid, hard-coded processes work well. As you get into later stages, if you're manufacturing something, you're probably doing the same thing over and over and over again. This works very well. But when you're working in research and development, and you're working with biologics, we are seeing our clients are changing their processes all the time. Not necessarily in big ways. They might not be overhauling everything, but they're changing, what kind of measurements they're taking. They're changing when they're running the assay. And this makes it really easy to break a traditional integration into a limb system. Another trend we kind of see are very specialized solutions that work really, really well for one particular kind of assay, or maybe one particular scientific group, but they don't work well across the entire organization. Every lab in an organization might be doing something slightly different, and the specialized solutions can't be generalized. So what we're seeing across all of this is that it creates bottlenecks for data aggregation and traceability, and ultimately it slows down the research. That's why we invested in Benchling for lab automation. We already know that Benchling is very well suited to be used in a wide variety of different scientific use cases, right? In a wide variety of different contexts. We have very large organizations deployed Benchling in all sorts of different labs doing all sorts of different things. So we're able to be generalizable. And then on top of that, what we see for Benchling for lab automation is it's centered around your notebook. And we're going to see a demo of this, and I'll show this in maybe 10 minutes or so. You design an experiment in the notebook. You tell Benchling when you would like to interact with a liquid handling robot or an instrument. And then Benchling is able to generate instruction files for those robots and instruments and ingest the output files that we get. So, a couple key benefits of this. One is this is all scientist-driven. So in the notebook, the scientists will be picking their input samples and their parameters, everything like that. A second big point is this is all point and click. What we're going to be seeing is all point and click. There are no, custom, scripts. There's no custom Python or analysis or anything like that that you need to run. Which means that if you change your experiment, it's fairly easy to change your automation tools to adapt to the new workflows that you're working with. After you generate your instructions, you give those to the robot. You give those to the instrument. You're going to get some output files back, right? And you want to be able to ingest those into Benchling. Likewise, this is all very easy to do. It can be done completely point and click. If you would like to write a script that automates that upload, so whenever a file comes off your instrument, it automatically gets uploaded into Benchling, you can write an API integration to do that, and there's a single API endpoint that you need to call. So you don't have to mess around with lots of back and forth, anything like that. And kind of the outcome of this is that you can create result tables in the notebook, new samples in your registry, and you can update samples in your inventory. So we released these tools several months ago, and what we've seen in the past few months has been really exciting. To start with, we've seen several customers use it for all sorts of scientific use cases, everything from hybridomal work to NGS. But the biggest use case we've seen is something that wasn't planned at all. And that was diagnostics. That was COVID-19 testing. It's become our most popular use case, and a lot of that has been because the work is very urgent. People really need to do it, and Benchling is, we've made ourselves pro bono for this testing. So over a dozen labs have been able to sign up and work with it. I want to kind of focus on one particular case study with Corteva. Corteva is an agriscience company. They are not in the business of diagnostics professionally, anything like this. But in early April, they reached out to us and basically said, we want to go from a system where we have no lab to a system where we're running thousands of tests a day. we really want to scale this up. We really want to contribute. Because our system is very easily configurable, because it doesn't require a lot of custom code, we were able to design a solution with them in a week, and we were able to configure Benchling for them and train the scientists on it in 3 days. As of this recording, Benchling has processed over 20,000 patient tests using a lot of this lab automation software. So what we're going to look at today is, how lab automation actually works. If you're a scientist, what do you actually do? So what I'm going to do is we're going to focus on a screening workflow, and we're going to focus on these 3 steps I have circled here: how to make plates for a transformation, how to generate and consume the output file of a OD600 assay, and then how to do the hit picking. All right. So let's see how we actually use the lab automation tools inside Benchling, and how you would do these to interface with both liquid handlers and with analytical instruments. So what we've got here in Benchling, we've opened a lab notebook entry. So for those not familiar with Benchling, this is sort of like the nerve center of your experiment, right? It's where you write down everything that you're going to do. It works a lot like a Microsoft Word or something like that. You can just insert new lines, type things. And what we have in this particular lab notebook is, here's the workflow that we talked about earlier that we're going to do. And we're going to see that we have some objects called runs. So these are for instrument runs, and we have 3 of them. And objects like this are how we are going to interface with liquid handlers and instruments. So we have a transformation run here first. This is to interact with the liquid handler to create plates that will transform vectors into strains. And then we have an OD600 run. So this is to interface with an instrument and collect results from an OD600 assay. And finally, we have a run for hit picking. So in this step, we are going to create more plates where we combine some strains that we transformed earlier with some media for purification. And the interesting thing about this is that we are only going to pick the strains that had a high enough OD from the previous step. So let's get started. So first, we're going to talk about our transformation run here. So what we have to do is configure this. We basically just input some parameters of the experiment. So first, we're going to pick our plates. So I have a set of 3 plates of vectors. In the real world, you might handle up to 100 or something like this for a single liquid handler. In this example, we'll just use 3. So we're going to transform the vectors here. We are going to transform that into these strains here. And then finally, we're going to pick a 3rd plate, and these are currently empty, where we are going to transfer both the contents of the other 2 sets of plates. So these are transformed strain plates. And then we're going to say that we're going to transfer 10 microliters of each. So we're going to save that. And as you can see, if I open up this plate here, this plate is just empty. It's going to be filled. And if I open up, this here, we're going to see a bunch of different vectors, one in each well of the plate, as you can see here. Cool. So we've set this up. Next thing we're going to do is click this Generate button. And when we click the Generate button, what it's doing is it knows that we want to basically stamp this plate and this plate into this plate, or stamp these 3 and these 3 into these 3. So it's just generating an instruction file for a liquid handler to carry that out. So let's open that up and see what it looks like. And I'll zoom in a bit here. So this is like a fairly standard kind of setup that you see. We got a column for the source plate, column for the destination plate. This is just a one-to-one stamp, source position, destination position, volume. And as you can see, we have this twice. We have this for all of the strains that we were using. This is that plate. 3 sets of strain plates. And then finally, we also have all of the different vectors, one per well. So these are the instructions. So I would, download this file, take it over to my liquid handler, upload that, run the liquid handler, and the liquid handler is going to produce an output file, right, to tell us what it did. Hopefully, that liquid handler's output file looks exactly like the input file does, because it just records what it did. So, exact same source plates, exact same destinations. Nothing went wrong. So what I'll do there is I will upload that output file. I am going to imagine that it's the exact same as the input file I just downloaded, because we're going to assume our liquid handler behaved correctly. So I'm going to import that, and I'm going to run this processing step. So what the processing step is the opposite of the generate step. The generate step is saying, let's take the data in Benchling and create liquid handler instructions based on it. The processing step is saying, let's take all of the information in this output file and carry out those actions in Benchling. So we have 6 plates total on the source that we are transferring into those 3 destination plates, and we can kind of see these 3 destination plates listed here from above. And if I open that up, we're going to wait a little minute, and this is going to populate here with, you can see 20 microliters, right, 10 from each of the sources. And if I open this strain, this is a transformed strain entity, you can see that we actually track the lineage here too. So this transformed strain has 2 fields, a source and a vector. And you can see that this is actually the vector that was in the corresponding well of the plate, and the corresponding source strain as well. So that's how we do lineage tracking there. All right, so I'm moving on to my next step. I now have 3 plates of transformed strains. I want to run this assay. So in the lab, I'll take that over to the instrument. I'll run the assay. I don't need to create any input file for this instrument. There's no special instructions I need to give it. The plates are already barcoded. Maybe I'm typing those barcodes in, but nothing fancy going on there. So the only step for the OD600 run is processing the output file. I don't need to generate an input file for this instrument. So I'll run the physical instrument. I'll get an output file, and that output file will look something like this, right? This is our example output file for OD. And it's just the plate barcode, the well, and the OD reading, right? And we can see here, plate 39, 40, 41. So I'm going to upload this OD data here. I'll process this. And this is doing the exact same thing we did in the previous step in terms of processing the result file. But whereas the previous step was making transfers in Benchling, right? The previous step was saying move, the contents of this well into that well. This step is creating results. So you can see that our lab automation tools are fairly flexible, right? We can take a number of different actions. We can create a number of different object types. So the first one was, creating those transformed strains. These are creating these results. So we can see all these strains from the previous step that were in previously, and you can see the OD reading for each one. These are the exact same readings that we see reflected here. And now what we're going to do is we're ready for hit picking, right? So we have 288 strains total. We want to pick the best ones. So for this, we're going to go to our Insights tool. We can go down here. We have a hit picking dashboard available. And what we can see here is we basically can set an OD threshold to be able to kind of see here all of the different strains that we just measured. I have this set up to just like pull data from the last hour or so. And we can change this OD threshold to whatever we want. So in this case, maybe I'll say we want OD 1.2 or greater. So what this will do is find all strains where we had an OD that was sufficiently high. The advantage of owning the demo data is we can make sure this will fit neatly into a plate. So we've got all of these. What we're going to do is add these to a work list. So we can have a work list called hit picked strains. So these are the ones that we want to do. Add these to the work list. And then when we come back here, we'll move on to our third step. So this is a third run. It works very similar to the other ones. We want to combine those strains with some media in a new destination plate. So for media, it's the same thing that we did similar to last time for the strains. We have a work list ready with a media plate. For destination, we're going to pick an empty destination plate here as well. So this will be purified protein plates. That's what we'll be doing here. But for strains, we're not actually going to pick a list of plates. Instead, we're just going to pick a list of strains. These hit-picked strains here. And I can kind of look at my list of strains, see that these are right 96 samples I want to do. So what I'm showing is that LabAuto isn't confined to plates. In the previous steps, we were sort of saying, take these plates in Benchling and stamp them into others. But we can also just list samples directly. We can say, take these 96 strains, find which plates they were in. These strains were not all necessarily in the same plate. Find what plates they were in, find the well, and then generate instructions for a liquid handler that uses that plate and that well. So we'll say here, again, maybe we want all of it, right? So there's 20 microliters of the strains, 20 microliters of the media. We will save that. It will generate our liquid handling file. And from here, we're looking at the exact same process we just saw in that first step, right? We see an input file, we'll download that and get the output. But what's nice to look at in this one is, whereas the first file was sort of a boring stamp, right? It was just A1 to A1, B1 to B1, and so forth. This one, what we're seeing is kind of a mix of these plates. We didn't order these, so this is just kind of going from all over the map. But we've got a couple hits that were in these wells in plate 39 and 41 and 40. So we can just kind of find these strains kind of from all over the place, or at least from the 3 plates that we were working with, and transfer those into one destination plate. Along with the media is much more orderly. We're just stamping the media right over. So that's a bit more of a complicated liquid handling step. So from there, we're looking at the exact same pieces before. We have our input file. We will upload the same as our output file. Import that. We will execute this processing step. And from there, we will have a single plate at the end that has all of these transform strains along with the media, which we can see here. And you can see, once again, we now have this purified isolate object, which will, once we open it up, link back to the strain that it came from, as well as the media it came from as well. And that's how you can run a experiment in Benchling that interfaces with both liquid handlers as well as analytical instruments. So please visit our virtual booth, and let's proceed to a Q&A session where I'll be able to answer questions. Thank you very much.